Muy buenas tardes tengan todas y todos ustedes. La Facultad de Medicina de la Universidad Nacional Autónoma de México les da la más cordial bienvenida al Seminario Internacional de Medicina Genómica a cargo de la Unidad de Investigación en Medicina Experimental. Presiden esta ceremonia la doctora Ana Carolina Sepúlveda Vildósola, directora de la Facultad de Medicina de la UNAM. El doctor Jaime Berunmen Campos, responsable de la Unidad de Investigación en Medicina Experimental. Y el doctor Roberto Tapia Conger, profesor de la Facultad de Medicina de la UNAM. Sean todas y todos ustedes bienvenidos. Para dar inicio, damos la palabra al doctor Jaime Berunmen Campos.
Hola, buenos días. Muchas gracias por estar aquí. Este es nuestro primer simposio internacional de este año. El año pasado hicimos 4 simposios y este año estamos planeando hacer 5 simposios. Es el primero y la verdad agradecemos mucho a la doctora Carolina Sepúlveda, nuestra directora, que nos acompañe y apoye el evento. Y a todos ustedes, traemos invitadas de mucho nivel, a la doctora Piper Bello, a la doctora Carrie North, que después las voy a presentar en más detalle con todo el simposio, pero darles la bienvenida y esperemos que este simposio sea de su agrado y a todos los que están conectados por internet también. Le voy a dejar la palabra a mi directora, la doctora Carolina Sepúlveda, que nos dé la bienvenida formal al simposio. Muchas gracias.
Muy buenas tardes a todas y a todos ustedes, a quienes nos acompañan de manera presencial y a distancia. Es un gusto para mí darles la bienvenida a este Simposio Internacional de Medicina Genómica organizado por la Unidad de Medicina Experimental de esta facultad, la cual versará sobre el papel de las ciencias ómicas, la genómica, la transcriptómica, la metabolómica, en el desarrollo de las enfermedades metabólicas, en especial la obesidad y la diabetes tipo 2, problemas de salud que son muy importantes en nuestro país, ya que ocupan los primeros lugares de morbilidad y mortalidad. Agradezco a la doctora Piper Bello, de la Universidad de Vanderbilt, y a la doctora Carrie North, de la Universidad de North Carolina, quienes son líderes internacionales en estas ciencias genómicas, por estar aquí con nosotros en este simposio. Espero que también tengan oportunidad de conocer nuestro hermoso país. Estoy segura de que su presencia en este evento será de mucho beneficio para la comunidad científica de la facultad y de otros lugares, dado que varias personas se encuentran vinculadas en este simposio a través de las redes sociales. La ciencia, junto con las actividades académicas y la difusión de la cultura, son los pilares de las actividades de la Universidad Nacional Autónoma de México y de esta facultad. Por esto, tenemos un gran interés en fomentar y apoyar este tipo de eventos académicos con profesores internacionales de gran prestigio, como nuestras invitadas el día de hoy. La ciencia en general y en especial las ciencias genómicas están creciendo exponencialmente y Estados Unidos, de donde proceden nuestras invitadas, se desarrollan a un ritmo más acelerado que en México. Además, son ciencias tan amplias y extensas que no se puede avanzar y competir sin la colaboración de muchos grupos de trabajo, algo que tenemos que fomentar más en esta facultad. Por esto, traer científicos internacionales periódicamente a nuestra universidad, como el día de hoy, nos permite primero tener la oportunidad de escuchar de viva voz nuevos conocimientos generados muy recientemente y segundo, estimular a los profesores y estudiantes, tanto de pregrado como de posgrado, el entusiasmo por descubrir, por generar nuevos conocimientos, por querer ser mejores y por competir. Me da mucho gusto ver tantos jóvenes en este auditorio. Y finalmente, y quizá lo más importante de tener en México a las doctoras Bello y North, es generar espacios y momentos de oportunidad para discutir nuevas ideas y proyectos de colaboración, así como planear estancias de trabajo de estudiantes y profesores en sus laboratorios en Estados Unidos. La convivencia cotidiana facilita la generación de buenas relaciones sociales y de amistad, que son la base de toda colaboración en grandes proyectos. Celebro entonces las actividades de este seminario y felicito a la Unidad de Medicina Experimental por dar continuidad a estas actividades, la primera de 5 este año. Muchas gracias y buenas tardes.
Y de esta manera damos por terminada esta ceremonia inaugural. Para continuar con los trabajos académicos de este seminario, haremos una breve pausa de 3 minutos.
Bueno, los 3 minutos ya pasaron. Vamos a empezar con el simposio. El simposio se llama Genómica. ¿Mande? Modo presentación. Sí. Ahí está. Genomics, Transcriptomics and Metabolomics of Cardiometabolic Disease in Mexican Population. ¿Por qué decidimos organizar este simposio en México? ¿Por qué es tan importante? Entonces, si ven la gráfica, estos son datos del estudio prospectivo de la Ciudad de México, que más adelante vamos a comentar un poco más, y el doctor Tapia y Jesús Alegre. Pero vemos en esta gráfica que en este estudio que se recolectaron o se reclutaron más de 150,000 individuos, y aquí yo muestro los datos de casi 107,000, el 20% de la población tiene diabetes o una enfermedad cardiovascular. Eso es lo que está en rojo, amarillo y naranja. Lo que está en azul son los individuos que tienen síndrome metabólico, o sea, que tienen 1, 3 o más de los 5 factores con los que se hace el diagnóstico de síndrome metabólico. O sea, que sumando los dos, más del 60% o casi el 60% de la población tiene diabetes o síndrome metabólico. Un verdadero problema en nuestra población. Por eso estamos muy interesados en estudiar la diabetes, la obesidad y el síndrome metabólico, y por eso organizamos este simposio con las doctoras Bello y Cari North. Fíjense aquí, los individuos, por ejemplo, que fueron diagnosticados con síndrome metabólico a los 3 años, porque estos individuos los hemos seguido 25 años. Se fijan aquí en la gráfica los puntos azules. A los 3 años, 4.3% de los que tienen síndrome metabólico desarrollan diabetes tipo 2. Y a los 25 años, el 22%. Pero no sabemos quiénes son los que van a desarrollar síndrome metabólico. Pero aquí calculamos el riesgo. Si tiene un factor positivo, 2, 3, 4 o 5 factores positivos, el riesgo aumenta exponencialmente. Entonces, estos individuos que ahorita los diagnosticamos con síndrome metabólico, pasan 25 años, claro, cambia el fenotipo, las influencias ambientales, pero muchos de ellos van a terminar con diabetes tipo 2. O sea, en el transcurso del tiempo pasaron cosas, pero algo traen también los individuos de base. La información genética, la transcriptómica o la metabolómica que vamos a ver el día de hoy. Entonces, los participantes en nuestro simposio, la doctora genético de enfermedades cardiovasculares. Lideré múltiples proyectos a gran escala en colaboración con consorcios genéticos internacionales. Es miembro del consejo editorial de múltiples revistas y funge como líder de la Obesity Society and the American Heart Association Epidemiology Council, y fue nombrada recientemente como directora asociada para la investigación en Gillings School of Global Public Health. Con ellas, gracias a el interés común que tenemos en las poblaciones latinoamericanas, a las buenas relaciones académicas que hemos establecido, vamos a empezar 2 proyectos muy pronto. Uno de ellos es el GTEx-MEX, que es un atlas de genotipificación y expresión genética de alrededor de 20 tejidos, de 200 cadáveres o más. Es importante, una vez que uno identifica las mutaciones, pues identificar si la mutación tiene un impacto en la función génica. El otro proyecto que vamos a empezar también con población del Hospital General es Molecular Underpinning of Cardiovascular Disease in Hispanic Latino Children. Ese proyecto, muy probablemente, si conseguimos el apoyo del NIH, que la doctora Craino y su grupo solicitaron, pues probablemente vamos a empezarlo muy pronto. La discusión la va a dirigir el doctor Jesús Alegre Díaz, que es miembro de la Unidad de Medicina Experimental. Él es médico cirujano, después hizo cirugía como especialidad, luego hizo una maestría en ciencias, en epidemiología, y actualmente él coordina el proyecto y es el investigador principal del proyecto del estudio prospectivo de la Ciudad de México, y él cofundó conmigo el área de epidemiología genómica de grandes poblaciones de la Unidad de Medicina Experimental. Muchas gracias, Jesús. Además, es un excelente guía de turistas que ayer nos enseñó casi todo el Museo de Antropología. Y finalmente, las conclusiones las va a dar nuestro amigo el doctor Roberto Tapia Conger, que es uno de los líderes de epidemiología de este país. Él es médico cirujano por la UNAM y después hizo una maestría en salud pública y en ciencias en la Universidad de Harvard, y un doctorado en ciencias por la Facultad de Medicina de la UNAM. Desde hace más de 46 años se ha desempeñado como profesor de la Facultad de Medicina, y la universidad le otorgó el premio al mérito universitario. Ha recibido diversos premios y reconocimientos a sus contribuciones en la salud pública, destacando el premio al mérito médico 2017 del Consejo de Salubridad General, y ha sido profesor visitante en la Universidad de California, en los campus de Irvine, San Francisco y Berkeley. Es miembro del Sistema Nacional de Investigadores Nivel 3 y ha publicado más de 200 artículos o publicaciones. Para destacar algunas de ellas, 2 libros: el Manual de Salud Pública en su 3ª edición y el libro de Salud Digital, que recientemente fue publicado por la UNAM. Él hace 30 años fundó el proyecto El Estudio Prospectivo de la Ciudad de México junto con el doctor Richard Petto, que es un proyecto donde se reclutaron a más de 150,000 individuos y que se han seguido en el tiempo y que ahora está dando una gran cantidad de resultados. En ese proyecto recientemente, hace 5 o 4 años, se secuenció todo el exoma y el genoma humano. En ese entonces él me invitó al proyecto y por eso estamos aquí trabajando en conjunto. Muchas gracias, Roberto, por estar con nosotros también. Pues sin más trámite, vamos a darle la palabra a la doctora Carrie North para que nos presente su plática, que se llama Molecular Mechanisms of Obesity Pathogenesis in a High-Risk Mexican American Population. Thank you very much, Carrie.
Okay, thank you so much for inviting me to be here today. It is my great honor to be here, and I'm just very much enjoying collaborating with the great group of scientists that Dr. Jaime, Dr. Jesus Alegre, and we're just having so much fun, and I hope to share some data from our studies that we've been working on to give you an idea about what can be done with multi-omic studies in maybe studies that you guys are working on. So today, I'll start with some background about the trait that I'm going to be talking to you about. Overweight and obesity, as most of you know in this room, are serious global health problems. Over 2 billion adults have overweight and 671 million adults have obesity. And by next year, it's predicted that over 1 billion adults will have obesity. Obesity has a tremendous impact on health. There are over 236 comorbidities affecting every organ system and medical specialty. The economic costs of obesity and its downstream sequelae are staggering. Estimates range from 147 to 210 billion years in the US every year. It's responsible for over 62 billion in Medicare and Medicaid spending. It also has a huge impact on loss of life. Each year, over nearly 3 million people die due to complications of overweight and obesity, which typically come from downstream sequelae, one of which Dr. Bello will be talking to you about, type 2 diabetes. As most of us realize at this point, obesity results from complex individual susceptibility and environmental factors. So there's many social determinants of health that play a large role, but we also have learned very recently and are beginning to learn more about the many molecular mechanisms that are underpinning obesity susceptibility. Some of them, many genetic factors have been identified, and I'll talk to you about that very briefly. But what we're really interested in now is taking that genetic data that we have, the loci that have been identified, and trying to partner them with other molecular information, multi-omics that I'll talk to you about today, in specific proteomics, to try and understand what the actual gene is doing and what the molecular mechanism of disease is. This becomes very important with obesity because we must realize that obesity is not probably one disease. It's probably many pathways of disease that we are together summarizing as one condition. The population that we are studying obesity in and that I'll talking to you about today is a Mexican-American population that lives on the border in Texas. They are self-identify as Mexican Americans, and they have a very, very high rate of obesity in their population. So these rather busy plots show you men on the one side, women on the other side, and this is from the United States data. And the yellow line, and I'm drawing your attention with the arrows, are showing you the non-Hispanic, the Mexican-American population. And as you can see, and this is across time from 1990 to 2018, there's a huge rise in obesity in the Mexican-American population. And if we, and this is plotting both at the top is obesity, which is a BMI of 40 or greater, but also a very devastating condition, severe obesity, 30 or greater for obesity. Severe obesity is a BMI of 40 or greater. And it's interesting that in men, Mexican Americans, the rate of obesity is rapidly growing, but in Mexican American women, you find that it's going down a little bit for obesity, but that's because severe obesity phenotype is rapidly growing. So it's a big problem in both men and women that self-identify as Mexican Americans. This is from a study that I work on called the Hispanic Community Health study of Latinos. And it's a large epidemiological study trying to understand the variability of populations that are living in the United States that come from different places in Latin America. And so it has individuals that self-identify being from Mexico, but also being from the Dominican Republic and Puerto Rico. So it's a really important study that we have ongoing in the United States. And this paper was recently published, and basically what we were doing is we were looking at excess BMI and correlating it with the prevalence classified by obesity. So you have men and women, and the rate of various cardiometabolic diseases, sequelae that we think are highly associated with obesity. And so as you can see, there's a very strong relationship with obesity for many of the phenotypes here that are what we would typically call cardiovascular disease, cardiometabolic diseases. Most striking and very striking is hypertension, and I just want to draw your attention to there because this factors into some of the data that I'm going to show you a little bit later. So what are the major drivers of these disparities? Why do we see some populations that have much higher rates of obesity than other populations? Well, certainly lifestyle, cultural norms, healthcare, access, psychosocial, socioeconomic, disadvantage, environmental toxins, all of these are going to play a very large role. And often it's a combination of factors. And our methods until very recently have only been able to look at one thing at a time. But we're now coming into an era where we are able to look at multiple factors at the same time. And this is really transforming the way we can understand disease pathogenesis. So why social and environmental factors probably play probably a larger role than some of the genetic underpinnings of these diseases. Susceptibility, some of that susceptibility is certainly influenced by genetic factors, which may be ancestry specific or have complex interactions with environmental factors. And we may see patterning across populations like what we do see. So I like to think of our genomes as a history book that has unbiasedly captured human experiences over thousands of years. And my question that drives my research is, what is that book teaching us about obesity? So how do we study genetic influences of obesity? There's been a recent, in 2010, there was a method that took us, the genomics community, by storm. It's called genome-wide association studies. And basically it was an unbiased look across the entire genome of individuals, querying genetic variation across that entire genome with the goal of identifying associations with any given measurable trait. So it's been used to study all kinds of complex traits, one of which is, of course, obesity. And so basically you have a sample, a very large sample typically, to have power to discover genetic effects. You extract DNA, you measure the phenotype, you do some association testing, and then we find regions of the genome that influence susceptibility to disease. And while we are very successful at identifying regions of the genome, we have much to learn about the actual functional polymorphisms underlying these signals, and that's what I'm here to talk to you about today. So where are we with our knowledge in terms of the genetics of obesity? And I'm here to tell you that we've come a long way than thinking about, black and white Punnett crosses. And, in the beginning, it was a bit of a perilous adventure to try and find underlying genes that influence susceptibility to complex traits. We had a lot of failures with our candidate gene literature, for example, but with GWAS studies, we have really transformed the science and we are really beginning to understand quite a bit. So what do we know about the genetics underpinnings of obesity? Well, Misa Graf and I, who's my colleague that's here, who taught a seminar yesterday, we work on a study called GIANT, which is a huge study of anthropometric traits across world's populations. We are working on a paper right now where we have mapped 6,000 different loci that influence susceptibility to obesity-related traits. So pretty much everywhere you look across the genome, you're going to find a signal that influences susceptibility to disease. These studies have also allowed us to understand a little bit more about mechanisms of disease, and I'm going to talk to you about our efforts in that regard. But one of the things that was very striking from the beginning, and what continues to be striking as we move forward in these studies, is that many of the time when we're studying overall obesity measures, BMI, not a great measure, but it's available in many, many people. And what we find is that many of the genes that seem to underlie obesity and overall obesity tend to be CNS focused, the central nervous system. So they tend to be genes that are involved with satiety and hunger. And even some would say, with some of the GLP-1 data that is coming around the corner, addiction. And so the motivation, the food motivation to eat. In contrast, when we look at genes that influence central adiposity, so how we distribute our fat, and when we distribute our fat in the central region, we find genes that are much more in adipocytes and with lipids, and so very distinct pathways, whether you're studying overall obesity or whether you're studying central obesity. So our understanding of obesity mechanism is very incomplete because we have these great data now, these great GWAS studies where we have these thousands of loci, but for many of those regions of the genome, we have not yet identified what the actual causal gene is. And so we consider this a major gap in the literature. So to meet this gap, substantial investments in identifying obesity genetic loci have been made. I just told you about the 6,000 loci that we've been able to identify with this large international collaboration. But now we're shifting to understanding the molecular mechanisms linking these loci with obesity. And one of the things that I'm very proud about the work that I do, and am I proud of my compilations here with you folks in Mexico, is that I feel very strongly that we need to have, there's major research disparities in the populations that are studied. And Hispanic Latino individuals are highly understudied. And I and Dr. Bilo and Dr. Graf have made it a very big part of our research careers to make sure that Hispanic Latino individuals are represented in these discoveries. So I'm going to talk to you about how we take genetic data, so those GWAS signals, and how we integrate them with other omics data to try and understand what the actual gene is. So what are omics? Omics are collective technologies that measure characteristics of a large family of cellular molecules, for example, genes, proteins, metabolites, and technological advances have enabled us the measurement of the entire ohm. So it's not only that we have technology advances that have enabled us to study all across the genome, but it's also become less expensive so we can start to characterize more people using this information. And if we think about the genetic studies and the GWAS studies, that's sort of the gene, that's looking just at the single nucleotide polymorphisms, which is really an estimate of what might happen. In contrast, integrating it with omics will really give us a better understanding of what is happening. So multi-omic studies enable mechanistic insights into inherent, which are genetic, and mutable, in this case, I'm going to talk to you about proteomic factors that impact obesity and downstream disease. So I'm going to just talk to you about one study that is very much in progress at this point. And it is a study that is led by my postdoctoral fellow, Mohammed Yasser, who's shown in the left side. And then it's co-led by the senior group to the right, which is Misa Graf, Piper, and myself. And our study objective was to characterize the obese proteome in Mexican-American study participants from the Cameron County Hispanic cohort, and to assess the causes and consequences of the obese proteome on downstream disease sequelae. So the study that we're working in, and it's such a pleasure to work in this study, is called the Cameron County Hispanic Cohort. It was established in 2004 by these wonderful scientists on your right, Joe McCormick and Sue Fisher-Hawk. They wanted to conduct population-based clinical research. So they randomly recruited population using a population-based longitudinal design. It's a rolling cohort, so the cohort continues to grow, and we continue to follow up at every 5-year interval for those individuals that we can get to come back. Right now, we're at about 5,200 individuals. It's growing. We're investing a lot of our energy into getting additional funding for this study. It's 94% Mexican-American, and it is an area with huge health disparities. 34% of the participants are uninsured and 26% below the poverty line. We are doing extensive phenotyping and rich archive of specimens, which has allowed us to do multi-omics characterization, not just at baseline period, but we're also doing longitudinal follow-up. And this is so exciting because we can actually study individuals as disease develops. So we don't have to we can look at a person who prior to they have type 2 diabetes, which Piper will talk to you about, and after they have type 2 diabetes, and we can get at cause and consequence of these genetic effects. We are very successful in recruiting in this population because the staff recruits from the community to the CRU, and the lab and the staff are all trained in research procedures and techniques and come from the community. So we've been very successful with recruiting. So why am I going to talk to you about marrying the genetic data with the proteomic data today? Proteins play essential roles in molecular pathways that contribute to excess body weight and regional adiposity distribution. Proteins may influence the development of obesity and may be perturbed as a consequence of obesity. And by that, I mean the inflammatory state, milieu of being obese can also have an impact on the proteome. So in contrast to the genome, which you're born with and you keep for your life, the proteome is can be responsive to environmental factors. Obesity is associated with many downstream disease sequelae, and proteins may mediate the effects that we see in the association with downstream disease. The method that we use is a panel called Olink. It's a proteomic platform that is very exciting. It offers high-quality protein biomarker discovery based on a flexible and scalable technology platform. Olink's platform enables high-throughput, large-scale analysis of thousands of proteins simultaneously, while using less than a drop of blood and maintaining exceptional levels of specificity, sensitivity, and dynamic range. And because of that, it's becoming a very popular tool to wed with genomic data. So we take this from the circulating in the data that I'm going to describe to you today. We're taking from the plasma, but you can do proteomic typing of any tissue type, and you might expect different tissue types to have different proteomic effects. In this case, we are using the plasma. These plasma-based proteomics measures were made in the CCHC data. There's a total of 2,921 proteins that we were able to study. And this preliminary data that I'm sharing with you today is in 254 independent individuals. It's actually 259. We use quality-controlled and normalized protein measures. One of the innovations of the work that we're able to do in the Cameron County is to study multiple obesity phenotypes. One of the things I mentioned to you was that BMI is not the best measure of obesity. But in Cameron County, we have more exquisite phenotyping of obesity. We can look at BMI overall. We can look at cutoffs or thresholds of severe obesity, for example. We can look at central adiposity measures, but we also have DEXA scan measures, which is an exquisite measure of obesity and fat and where it's distributed. So if we look at the 259 individuals in our sample from Cameron County, you can see they're on average about 50 years old. The phenotypes, the 13 phenotypes that we considered in this work are shown here. As you can see, there's a high burden of disease. This shows this slide I wanted to put in because it does show why looking across these different measures of obesity can be useful. So this is what we would call a phenotypic correlation. So you're looking at all of the traits that we looked at and how correlated they are with one another. And so you can see for some traits like waist-hip ratio, the correlation with other obesity-related measures is quite low, and it could give us additional information. You might also see that if you have BMI, it highly correlates, but there's something that we're missing by not looking at more exquisite measures, which is why we look across measures. So what we did in our data was we did a differential abundance analysis, and it's performed using a mixed model approach, which means we're allowed to account for individuals that are related. The models were adjusted for age, sex, principal components of ancestry, and the first 10 peers of peer factors at a minimum. And so the peer factors just help us explain unexplained noise in the data. We also looked with waist-hip ratio with the central measures of obesity. It's important that you adjust for BMI. It's also important that you look across sexes, which we did because there's distinct effects on central adiposity measures in males and females. And we controlled the false positive rate of the fact looking across all of these traits and all of these roughly 3,000 proteins that we looked at using an FDR correction using Benjamin Hochberg method. So this is what we get from our differential abundance analysis looking at a protein. This is what we call a volcano plot, and you'll probably see some of these in Piper's talk as well. But basically, this is looking at over on this right-hand side are individuals, are proteins that are more highly expressed in individuals who are, in this case, I'm showing you the severe obesity plot. And over here are more factors or proteins that are less abundant in individuals who have severe obesity. So this side of the pie would be giving us those protective factors, and this side of the pie would be giving us the at-risk measures. This protein up here, this highly significant protein, is one you might all be familiar with, leptin. We know this is a great true positive because we know that leptin has a very strong relationship with obesity. So this very busy plot that you probably can't see, but if you get a hold of the slides later, maybe it would be of use to you. But this is looking at across all the traits that we looked at, what the differential abundance analysis looked like. And in total, we identified 122 proteins that were associated with adiposity-related traits, and leptin happened to be the encoded protein that was the most significant across all the traits that we looked at, not surprisingly. So what we wanted to do, this was in a very small sample in Cameron County, and what we wanted to do is say, okay, now that we have these associations, what does it mean? What does it tell us about the underlying mechanisms of obesity? So the first thing that we did is we replicated our study findings. It's a key to doing omics research. And in this case, we had a wonderful database that happens to have 40,000 proteomic markers available to any researcher that wants to study it. The UK Biobank, they've used the Olink platform. We are considering here the maximally unrelated sample size of 20,000. So it has up to 40,000, but we use the individuals, every individual in our sample was unrelated to the other people in the sample. And we assessed the association of these top markers that we saw, the 122 markers that were significantly associated with in the UK Biobank. Did we see those associations as well? And excitingly, 118 of 122 of those proteins displayed directionally consistent and nominally significant associations with BMI in the UK Biobank sample. So this is an astounding replication. And this is also in a population that has different heritage than what we see in our Mexican-American data. So this suggests to us that there is huge generalization of proteomic effects for obesity across populations. The next thing we wanted to do is try and get at, can we find, using statistical tools, can we try and map out what is a cause, do these proteins actually go in the causal pathway towards obesity, and what are the proteins that are actually a consequence of being obese? So you are in an obese state, that might dysregulate your proteome. And so we use a statistical tool called Mendelian randomization that allows us to make inferences about cause and consequence. It does not demonstrate cause or consequence directly. It is a statistical tool to do this. However, it gives us great hypotheses, prioritization of proteins to carry forward for actual functional studies in mice, in vitro, et cetera. How does Mendelian randomization work? It harnesses Mendel's laws of inheritance. The segregation of alleles separate at meiosis, and a randomly selected allele is transmitted to the offspring. And so there's independent assortment of those alleles. So if we study the genetic variants that predict the exposure instead of the exposure, then you're born with those genotypes, so we can assess there is a causal relationship. We can look at it in one direction towards obesity, but we can also look at bidirectionally. So we can also look at genetic variants that influence obesity and then say, does it cause dysregulation in the proteome? So two-sample MR helps us understand potentially causal and consequential relationships between obesity and the circulating proteome. And this is the model that we used and the questions that we asked in our data. Is the SNP associated with protein abundance? And this was all done in using large publicly available data, genome-wide data. The second question, is protein abundance associated with obesity traits? And then is a causal relationship supported based on Mendel's laws? So to do Mendelian randomization well, you need very large sample sizes. So the sample sizes that we use for MR are indicated here, as long as the reference population and the different GWAS for the various traits that we looked at. The next question that we asked is, if we do find proteins that are associated causally or consequential with our proteome, do those same proteins cause or have a causal effect on many of these diseases over here that you see that are known for their relationship with obesity? So these are well-known, what we consider obesity-related conditions, or have a strong relationship with obesity. And so then we could ask, using the proteins, as opposed to just looking at obesity, yes, no, do these proteins actually have a causal impact on these diseases as well? So the genome-wide association studies that we used for those data are shown here. And one of the things that you'll notice is that the sample size needs to be very large to do Mendelian randomization well. So what did we find when we looked at our data, when we looked at the Mendelian randomization? We found several different patterns in the data. We found some proteins that seemed to have a causal effect on obesity. We found some proteins that seemed to have a consequential effect from obesity. And we took any kind of causal or consequential and asked, do they have a causal effect on those downstream diseases that I showed you? And we found that 101 proteins, we had MR evidence for a causal relationship, 63 had a causal influence on obesity traits, 45 seemed to be dysregulated as a response to obesity, and 66 proteins taken from those 63 and 45 had a potential causal effect on these downstream obesity-related diseases. So this very busy plot is showing you the traits that we looked at on the bottom, like the body fat percentage, BMI, et cetera. And these are the proteins that we found causal relationships with. Across the top are those obesity-related diseases that I showed you. One of the things that I think is striking is, and I've done this by coloring, is we find the red is showing you proteins with a causal influence on obesity and downstream disease. The green is showing you proteins that are dysregulated by obesity and with a causal influence on obesity and that downstream disease. And the blue are showing you proteins that cause obesity dysregulation but also influence downstream disease. So one of the things that's very striking about this figure is that across the different measures of obesity, we may not always see the statistical evidence for the causal relationship, but you certainly see the clustering across the different traits. So you don't have one trait showing a causal and another trait not showing a causal, or one that has a causal versus consequence. They seem to be consistently either causal or consequential. And because of that, for the rest of the talk, I'm not going to spend a lot of time about the different traits, but I'm just going to show you some of the other data, assuming that these patterns are similar across these traits. So the next thing we wanted to do, that gave us a great, thank you. So that gave us a great understanding of traits and diseases that we knew were probably related to obesity. But we also wanted to look across the entire phenome to see, are there relationships that we might not expect? Are there relationships with disease? Or does obesity have relationships with disease that we might not expect? So we used an approach called PheWAS, and we used that same resource, the UK Biobank data, and we took all of the proteins that we were interested that demonstrated causal relationships, and we looked at the relationship with downstream disease all across the phenome of the UK Biobank. So this is a sample of 500,000 individuals. And so each protein individually, the 100 or so that we found with interesting relationships with obesity, we looked all across the genome and across a variety, see if I can do pointing, of different classes of traits. There's probably about 500 different traits. So I can't possibly show you all of them. Each of these dots are those different traits, but we classify them according to overarching types of disease. And we asked the question, do proteins with causal and consequential effects on obesity also associate with downstream diseases phenome-wide? So we used the UK Biobank. We took 101 proteins that had a causal influence, and we summarized the PheWAS in the next couple of heat plots using the coloring scheme that I had shown you earlier, but looking at those that were causal to obesity, those that were dysregulated because of obesity, and those that demonstrated both a causal effect and an effect of dysregulation. And we asked for those classes of proteins, what diseases do we find them associated with? So to reduce the dimensionality in the next slide that I'm going to show you, we take those 500 plus terms and put them into about 38 different classes. We selected the most significant association of each protein with each disease spectrum. And then we created these heat maps where rows represent proteins and columns are diseases across the disease spectrum. The red color is going to represent positive association, and the blue color in these heat maps is a protective effect. So just to give you an example of what I was just saying, these are the different associations. These are different genes that we looked at. They're associating with type 2 diabetes, diabetes mellitus, but here we put them into a spectrum group that we call diabetes or that we call obesity, et cetera. So that's where we came with our classifications. So this heat plot shows you across the bottom some of our significant findings. And the coloring, again, this red means that there's a strong increasing obesity effect, and the blue shows us that it's a protective effect. And each row is a protein, and each down here across these are all these different classes of obesity, of disease traits. And what we find is, strong relevance of the phenotypes that we expect, cardiometabolic diseases, et cetera. But we also do see other things showing up for other traits that we might not expected. So we're just beginning to dig into this data, and it's very exciting. This particular plot shows protein abundance that has a causal influence on obesity. This is showing you protein abundance that is influenced by obesity and both caused by obesity. So this is both a cause and consequence. So the dysregulation of this protein is both a cause and a consequence of obesity, and it associates with diseases here shown in the coloring, with the reddish here, so it's having a somewhat modest effect on diabetes, but not a strong effect over the other the diseases. But when you look here, the bright coloring are the very strong effects. And this is where protein abundance is dysregulated by obesity. So one of the things that I wanted to draw your attention to, and relating it back to one of the things that we're very interested in now, is that the very strong relationship we're seeing in the proteome with hypertension-related diseases. It's much more than we'd expect by chance, and it's both for proteins that are causal and consequential. So we're really interested now in developing a research study to look at this relationship between hypertension, renal disease, and these proteomes that are associated with obesity. We are just digging into this data, so I don't have any good answers for you yet, but this is something that is very striking in the data. So this hopefully will show you a study where we're wedding together genomic data and omic data to try and better understand the obesity mechanisms of disease. Our study, I think, addresses many research gaps, the need for proteomic studies to refine our understanding of what these BMI genetic loci do, the need for additional omics data to link genetic variants to causal genes, and importantly, looking in a high-risk population. So in the future, what do we hope to do with this data? Proteins robustly associated with obesity and downstream disease sequelae should be considered as potential drug targets and as biomarkers for early disease risk prediction, and not just the proteins, but the proteins that the pathways that these proteins are in. We replicated our data in a very large sample, and the replication suggests that there's a very large generalization at the level of the proteome across populations. The patterns of association across diseases need to be further characterized and we see with those heat plots very distinct patterns that we need to understand now and unpack. We will unpack that, especially in the next month. We would plan to have data delivered to us from the Vanderbilt lab in about 1,000 additional individuals from this same data set. So we're extremely excited for getting those data. And so I'll end with acknowledging that team science is extremely important in this kind of work. And this is just showing you a picture, a little segment of my lab from a much younger me. But this is a lab that I've been doing. I've been at this game for 23 years, and I'm very proud of the lab that I built and the many very talented individuals that work with us on this study. So I'll take any questions. Oh, I won't take any questions.
Bueno, muchas gracias. Thank you very much, Dr. North. We have our next presentation by Dr. Jennifer Below, Omics of Type 2 Diabetes in High-Risk Mexican-American Population. Thank you very much.
There we go. All right, thank you so much for having me. It's such an honor to be here with you all today. And thanks again to our wonderful hosts who've been showing us such an incredible time. Your country is so beautiful. It's been amazing to get to see parts of it. And yesterday we got to go to the Anthropology Museum, and it was overwhelmingly exciting. It was just so cool. So I'm here to talk to you a little bit about some early results. So these are very much hot off the press. There's a lot of questions that remain unanswered here, and I'm excited to go on the journey with my collaborators, Dr. Carrie North, but also the collaborators here from UNAM as we continue to unravel these questions. As I think everyone in the room is probably pretty aware of, type 2 diabetes is a major and growing health epidemic. Type 2 diabetes is the clinical endpoint of deterioration in glucose metabolism, leading to elevated blood glucose, which in turn leads to many sequelae and downstream disease. Approximately 422 million adults have type 2 diabetes, accounting for more than 4 million deaths annually. And in the US, 30 million adults have type 2 diabetes, but this isn't distributed across populations equally. We observe many health disparities in which specific racial and ethnic minority communities, in particular Hispanic and Latino populations, are adversely affected by type 2 diabetes. The increasing prevalence of type 2 diabetes and the fact that 1 in 4 individuals are actually undiagnosed underscores the need for not only novel diabetes prevention, but also better diagnostic and treatment strategies. Now, the same tool that Dr. Carrie North just told you about, this genome-wide association study, this has been deployed across obesity traits. It's also been deployed broadly in type 2 diabetes to improve our understanding. We have uncovered through these genomic studies in populations hundreds of variants that influence type 2 diabetes risk. And yet still, as she brought up, significant gaps in the translational pipeline remain. In particular, it's very difficult to understand what the genetic signature of this trait is in individual populations when all we tend to study overwhelmingly are European ancestry individuals. To give you some perspective, I am the leader of the Hispanic portion of the type 2 diabetes research group called Diamante. This is an international group comprising leadership that is taking responsibility for performing genome-wide association studies and meta-analyses of type 2 diabetes in European ancestry individuals, African ancestry individuals, East Asians, South Asians, and Hispanics. For perspective, we've recently published the largest paper that encompassed 2 million European ancestry individuals, but our Hispanic study only comprises 60,000. And at the time that we amassed this collective data set, it was all the data available in the world. I'm really proud to say that we've begun to collaborate with the individuals in this room who have been working over the last 20 years and have recently acquired genetic data mapped to the phenotypic information from 140,000 adults from Mexico City. This is going to transform our ability to understand these diseases. And it's really an honor to be in the presence of people who are doing such great work. So studies like the MCPS are going to go a long way to address this disparity in the populations. But even still, 140,000 doesn't touch 2 million. We have a long way still to go. But GWASs, as Carrie brought up, are not actually perfect in and of themselves. They do not uncover the actual causal effects of linking to disease. They tell us regions of the genome, but those regions may encompass many functional factors, many genes, or even worse, no functional factors or no genes. And then you say, why is this coming up in my study? So understanding the actual variation that is leading to disease is incredibly important. Further, in genome-wide association studies, the burden of multiple test correction is so high. We're testing millions and millions of genetic variants. And this means that we have to have these exceptionally large samples in order to be able to uncover disease risk. And furthermore, the effect sizes per SNP in genome-wide association studies tends to be very, very small. And as I just mentioned, the association signals can span multiple genes or no genes at all. So some people have worked to develop at Vanderbilt ways to leverage gene expression data, marrying that to the genome-wide association study data to be able to improve our understanding. So if you think about what influences a gene's expression level, it might be factors that are relating to your environment. It could be something temporally regulated. Maybe you had 7 cups of coffee this morning and that's changing how your body is processing DNA. But some portion of this pie that's regulating how a gene is expressed, the abundance of transcript that it's making, is genetically regulated. So I show you this plot to emphasize the fact that the portion that is genetically regulated may vary by gene, but also by tissue. So we have this set of influences affecting every gene in every tissue, but the proportion of the variance of the transcript that is explained is going to be different by gene and by tissue. But for those genes in certain tissues where there is a large component that is genetically regulated, we could endeavor to potentially infer or impute the abundance of the transcript from the genotypes themselves, rather than having to do the expensive and time-consuming process of performing the transcriptomic analysis. So this has led to the development of a tool called PredictScan, which was an invention of some of my colleagues at Vanderbilt and the University of Chicago, which allows us to impute transcript abundance from genetic data. So if you imagine that this is a cartoon of a gene, you've got the exons shown in the blue box, and you've got SNPs that are measured wherever I've shown you an arrow. You then, in the same data set where you have those genotypes measured, you also measure the abundance of the gene product, in this case, the transcript level, in a particular tissue. Here I've highlighted in orange the SNPs that are associated with the expression of this gene. So you do an association test that estimates the predictive value of that SNP for that gene's transcript abundance. And then what you can do is create an additive model that aggregates the evidence from all of the set of SNPs that are contributing to the gene abundance estimate into a single statistical model. So basically now we have a statistical model that allows us to start with genotypes and infer transcript abundance. So this has been done for about 50 different tissues from the GTEx project. I will note that the GTEx project, like so many resources that we have in human genetics, is predominantly European ancestry individuals. In fact, I think that in all of GTEx, there's either 1 or 2 individuals who identify as Hispanic. So another thing that our group is working on doing is trying to remedy that situation. Dr. Jaime Berumen mentioned earlier that we're trying to create this GTEx MEX data set, which would help to ameliorate this problem. So what does this allow us to do? We have these statistical models. It means that I could take a DNA sample from Dr. Alegre, I could take a DNA sample from Dr. Berumen, I could take a DNA sample from myself, I could measure the genotypes at these different points in the genome, and then I could infer from those genotypes what I would predict our transcript abundance to be. And again, as a reminder, this is not necessarily going to reflect the true transcript abundance. We're only estimating that genetic component of the transcript abundance. But once I have this estimate of the genetically regulated expression level, then I can correlate that to disease measures. So for example, fasting blood glucose or HbA1c as an example of linear or quantitative measures, but also of course I can do it in a logistic regression for type 2 diabetes. So we performed this using the results of our Diamante study, which allowed us to identify 65 genes that were associated significantly in terms of their genetically regulated expression with type 2 diabetes. 19 of these genes had been previously reported in the literature in genome-wide association studies as sort of the mapped gene that was hypothesized to be causal. So now we have additional evidence of causality mapping us to that gene. And then another 35 genes were in close proximity to the genes that had been previously mentioned in the literature, but were not the gene that signal had been previously attributed to. So it suggests maybe the GWAS was pointing us in the wrong direction. Maybe that signal was actually about a different gene. And it also helped us by aggregating these SNP effects. We actually reduce our multiple test correction burden, overcoming one of those challenges of genome-wide association studies. And it allowed us to identify 11 potentially novel genes that have either never been mapped in GWAS signals, nor are within a megabase of any previously reported variants. So this is one way to be able to sort of infer omics information from genetic data itself. And so I encourage you, as you're thinking about next steps for the MCPS study, that you could think about beginning to aggregate your single variant effects and orient them functionally around these gene units and hopefully improve your power. Now, this kind of inference comes with many limitations, notably the fact that we don't have great expression, an expression atlas in Hispanic populations, the way we do for Europeans measured in GTEx. And so it is important to consider what omics need to be measured and how the actually directly measured rather than inferred omics could help us learn more about type 2 diabetes risk. So I know you saw this slide and carries a lot. So we've done a great job of capturing genomics, but in this study, I'm going to tell you more about the directly measured transcriptomics, proteomics, and metabolomics and preliminary data that we've generated in the same cohort that she described, this Cameron County Hispanic cohort, looking at how these multi-omics all together are pointing to particular genes in blood as being associated with type 2 diabetes. So when I tell you about these studies, I want to bring up the fact that transcriptomic studies, as well as proteomic studies and metabolomic studies, are extremely understudied so far. These are emerging omics measures. These are emerging ways to think about measuring disease biomarkers. And so this is a study from 2022, where you can see that this is a review paper of all of the transcriptomic studies of type 2 diabetes to date. And these sample sizes are like 5 versus 7, 10 versus 10, 20 versus 30. These are tiny, tiny studies, really highlighting the research gap for actually doing this in a large scale and well-powered way. So Carrie and I have endeavored to collaborate on sort of a global study, multi-omics of health and disease, that is anchored in identifying cases and controls, measuring the phenotypes across time, as well as the omics across time, including genomics, epigenetics, metabolomics, transcriptomics, proteomics, lipidomics, looking at the microbiome and trying to do a really robust job of characterizing the entire environmental suite of variables that can affect disease. I'm sorry, this is a little bit cut off, but the goal then is to perform differential abundance analyses, where we look to see is there differential abundance in cases and controls in these different molecular measures. On the top, that's cut off, was the concept of doing Mendelian randomization to infer causality, then to use local ancestry, QTL analysis and familial analyses, as well as pathway analyses and structural equation modeling, to be able to integrate all of these molecular features to be able to profile the risk factors for disease. This is a slide that you've also already seen from Carrie, but just to remind you who the Cameron County Hispanic cohort, who the drivers really are, it's actually these study coordinators from the community. These are the individuals who are going out house by house, knocking on the doors. They are all members of the community, and this is really what has made this cohort successful, has been the work of those women. In addition, all the lab staff are also from the community and trained in the molecular procedures. And so to emphasize, 51% of the Cameron County Hispanic cohort are obese. Nearly 30% have type 2 diabetes, and about half of those individuals did not know that they had type 2 diabetes when they came into the clinic. A further 32% have prediabetes, nearly 30% are hypertensive, and 35% have metabolic associated fatty liver disease. So this is a population that is extremely disproportionately impacted by metabolic diseases. For this study, we defined type 2 diabetes by the ADA 2010 criteria, but we also classified somebody as diabetic if they were taking a diabetes medicine, or if they had been previously told by a doctor that they have diabetes, or they were previously diagnosed. This gives you a sense of the sample sizes that we have. They're a little bit bigger than 5 and 6. They still might not be as large as we would like to have. And these, as Carrie mentioned, we have another 1,000 proteomics measures from this population coming in the next month, which we think is going to be really tremendously exciting. You might notice that the transcriptomic numbers are a little bit lower than I just was previously mentioned, that they were in the 1,000. This is because for this study, we dropped out anybody with prediabetes. So these are diabetes cases and controls, with excluding anybody with prediabetes. We performed a cross-sectional approach to this analysis, where we had the blood and serum collection. We used omics platforms for data generation. We have phenotyping for type 2 diabetes, as I just described, and then we performed differential abundance analyses to be able to look at the molecular biomarkers that were associated with, where their increased abundance was associated with risk versus their decreased abundance was associated with risk. We used linear regression in a cross-sectional model to look at how the omics expression, or the omics abundance measure, was associated with these different variables with type 2 diabetes as a the effect estimate from type 2 diabetes was our variable of interest. After controlling for age, sex, the global genetic ancestry components, and then, as Carrie mentioned, surrogate variable analysis is a tool for being able to capture unmeasured variation that might contribute noise in your data. Okay, so some results. What we found was that in measured about 16,500 transcripts abundance, we found almost 750 genes that were significant after multiple test correction. And these include a wide range of genes that have been previously linked to type 2 diabetes in the literature. We also added a variable into our model of BMI because we're interested to understand, take the full set of transcripts that were associated with type 2 diabetes and then partition them into those transcripts that were associated with diabetes as part of a BMI mechanism or a weight mechanism, and those that are independent of weight but are still impacting type 2 diabetes risk. And what's interesting is that our top signal, ABCG1, remains top signal. Same with KLF10 and CPT1A. These all remain significant even after correcting for BMI, suggesting that there's a different mechanism that is associating these to type 2 diabetes risk. This is a full list of the top genes that were differentially expressed in type 2 diabetes. And we then sought replication because, of course, as Carrie mentioned, discovery is nothing without replication. And I was really gratified to see, and this is pretty incredible, that ABCG1 remained our very top signal. And again, with the same direction of effect, where less ABCG1 transcript was associated with type 2 diabetes. And this was true both, again, in the model that did not account for BMI and the model that did account for BMI. So these are highly concordant and consistent findings in our discovery and our replication. This little Venn diagram summarizes the main analysis, the discovery analysis, and the replication. But here in the Venn diagram, I'm showing you the set of genes that were genome-wide significant in the main analysis and the set of genes that were genome-wide significant in the transcriptomic replication. We find 93 genes that overlapped. However, if that's sort of excessively stringent criteria to deploy for a replication analysis, so if we only correct for the 749 genes that were tested, we actually find that 239 of our 749 genes replicate at a level of statistical significance. This is nearly a 33%, a 1/3 replication rate compared to the 5% that's expected under the null. So this is a really profound rate of replication in, again, sample sizes that by most measures would be considered small. So what is this ABCG1 gene that came up both in the discovery and replication as our top hit? It's a binding cassette subfamily G member 1. And interestingly, this gene has been previously associated in a GWAS of Scandinavian individuals with severe insulin deficient type 2 diabetes. So there's some prior evidence suggesting that there's a mechanism underlying this relationship that we observe. We also then carried out our differential protein abundance analysis. So the same models as I showed you before. And here we can see that we identified in a smaller, the Olink platform queries about 3,000 proteins. So in those that passed QC, we found 348 significant proteins by FDR, and some of them are shown here. And you're going to see in this plot probably gene names that you're pretty familiar with, including LPL and PLXNB2, many genes that are very familiar to people that are active in type 2 diabetes genetics research. I want to call out the gene GCG. I think this one is of particular interest. This is actually the precursor gene to GLP-1, which is, of course, the target of the new class of obesity targeting drugs. Ozempic, Wegovy are GLP-1 targeting drugs. We then sought to look to see if we had convergence of evidence coming from the transcriptomic and proteomic analyses. And I again want to stress the fact that this is an over-conservative estimate because we're basing this on genome-wide significant results from the transcriptome and genome-wide association in the proteomics. And so this is probably an over-stringent criteria to have to meet, but we identified 13 genes, including LDLR, again, which is one that you would very much expect to see, and some other favorites from type 2 diabetes research showing up in this convergence. And this is just highlighting those that remained significant in the overlap after correcting for BMI, suggesting that these genes are independent of the pathway of obesity as a causal factor for type 2 diabetes. I don't want to spend too much of your time going over the very specific details of these, but again, we'll make the slides available to you. So if you're interested in following up any of these genes, you can. But suffice it to say that many of the genes that we found have evidence of being involved in diabetes and metabolic diseases. For example, this AOC3 has been previously associated with diabetes and metabolic disease. CD93 was associated with leptin levels in a Hispanic population. LDLR is a well-known gene that has been associated with diabetes, HDL, LDL. EPHA4 has been significantly associated with hypertension, going back to, again, Carrie's findings in hypertension in human female participants with type 2 diabetes. And across the board, we're seeing many genes that make a lot of sense as being sort of functionally dysregulated as either a cause or consequence of type 2 diabetes. Finally, and this is going to be just hot off the press preliminary results from our metabolomics abundance analysis. So similar to what I showed you before, we looked to see which metabolites were differentially abundant in the blood and associated with type 2 diabetes. And we were somewhat gratified to see that glucose was the very top signal there. And we're still doing some work to try to understand if these metabolites are mapping back to regulatory pathways that are defined by the proteins and transcripts that we see as being differentially abundant as well. So you remember I spent some time telling you about the PredictScan analysis. So the PredictScan analysis results that I showed you, those 65 genes, again, that was an over-conservative definition of significance. That was an FDR p-value of 0.01. Here we've expanded that out to an FDR p-value of 0.05. And we're really interested to see where the genetically regulated expression level is for a gene is associated, the transcript abundance for that gene is associated, and the protein level is associated. But what you see here is that we actually don't have anything in the center of that Venn diagram. None of our genes met all three criteria. Now, I hypothesize that the reason for this is that the TWAS analysis, this PredictScan analysis at the bottom, was done in the wrong population. It was done with data from predominantly European individuals. So my lab has been working with gusto on generating EQTLs for this same Hispanic population. And my talk last year, I gave a lot of the preliminary data from that. We are now building those models and deploying them in this data. And we have actually also provided them to the MCPS so that when they do their transcriptome-wide TWAS or PredictScan analyses, they can also use Hispanic-specific prediction models. So for future directions, we would like to use the same approaches that Carrie described to you for Mendelian randomization and causal inference to begin to partition out which of these genes, the transcript level and protein levels, are associated with risk of type 2 diabetes and have a causal effect on type 2 diabetes versus those that are dysregulated by the state of having type 2 diabetes. We would like to do more integration of the metabolite data into the pathways that we're observing from the transcripts and the proteins. And we would also like to do some more work in gene set enrichment analyses, which currently are confounded because the proteomics data are actually, omics is a little bit of a misnomer there because it's a selected panel of 3,000 proteins. The new data that we're going to get expands that to 6,000 proteins. So it's a little bit better, but still selected because we don't have the full suite of 20 or 25,000 proteins assessed. And again, we would like to replicate our proteomic findings in the independent data that are forthcoming. And then once we have a prioritized list of genes that we believe are part of a causal pathway for type 2 diabetes, we would then want to partner with individuals who have expertise in functional biology and validation in model systems and cell lines. So with that, I'll close out. Thank you very much, especially to Lizzie Frankel, who's a very early career graduate student. I think she just completed her first year she just completed her second year in our PhD program, and she did all of this work. So really incredibly impressive work from Lizzie Frankel. And she collaborated very closely with a professor that works under Dr. Carrie North, Heather Hyland, who is also leading this work. So much credit goes to both of them. And also I'll throw out there that my lab is hiring postdocs. So please reach out to me if you're interested in coming to the United States and doing a postdoc at Vanderbilt. And with that, I think Carrie and I can both take your questions.
Pues muchas gracias a la doctora Piper. Thank you very much, Piper. A lot of information, as I expected. Pero como vieron, una gran cantidad de información que traen las doctoras americanas, información nueva realmente que prácticamente sacaron del horno estas semanas. Thank you very much. A lot of information, very interesting. Vamos a decir al doctor José Alegres que venga a dirigir la discusión. Vente, Jesús.
Oh, okay. I can translate the question, don't worry.
Okay.
Bueno, la verdad es que es un placer tener investigadoras de este nivel de Estados Unidos, como dijeron al inicio. Estas personas llevan millas adelante de nosotros en un tema que va a ser el futuro de la medicina. Nosotros vamos a tener marcadores genéticos que van a definir cómo nos van a atender nuestros médicos. No va a ser la misma medicina para todos, no va a ser el mismo tratamiento para todas las personas. Y por supuesto, las personas que estamos en mayor riesgo de algunas cosas, vamos a tomar algunas prevenciones muchísimo antes de padecer la enfermedad. Eso va a empezar a disminuir las enfermedades crónicas. Creo que en general está muy, muy claro el comentario. Si alguno de ustedes tiene una pregunta para la primera o la segunda. Sí, doctora, por favor.
Gracias. Great talk. Both of them were really nice with the data that you shared with us. The first question is about the proteins that you found that are differentially abundant. Were you able to classify them in clusters of a certain pathways, and which pathways are those?
So excellent question. And we are looking, there's the bias in the pathways that are represented on the O-link panel. So we can't give statistical evidence for whether this pathway is overrepresented, but we can dig into what pathways these proteins are involved in. And so that's the very careful work that I was mentioning that we're trying to do now by disease to try and make sense of what those data are telling us.
Yeah, to say that a different way, the O-link closer. Okay, to say that a different way, the O-link platform, they selected proteins for their assay involved in inflammation. And so when you put the list of signals that you get into any pathway analysis, you get inflammation, but that's because of the bias on the platform, not because that's a real finding. And there's other pathways that are interesting for metabolic disease that are not represented on the platform. And so because of that, if you don't find a pathway to be enriched, it's not a sign that there was no evidence from that pathway. It's a sign that that pathway was not measured. And so that's why it takes more nuanced approaches to uncover the meaning behind the proteins that we see than to just stick them in an analysis and press go. So yes, but we need a little bit more time.
So that could be the second question was that I was looking for the normal GWAS genes like TCF7L2, FTO, all of those, and they didn't appear in your protein. So it's the same reason, because you are limited by the O-link that you don't find them in those proteins.
That's right. Those are not measured on the O-link platform, but I'll also note that the transcriptomic is transcriptome-wide sequencing. So that is much less targeted than the protein. And we don't see those coming up in that data either. And I think that this might speak to some of a bit of a paradox that we do see when we study Hispanic populations from very poor communities in the United States. In the genetic analysis, we don't see TCF7L2. And it suggests that TCF7L2 might be working through a mechanism that involves greater potential variation in social determinants of health. We see a lot of SLC genes coming up. I would have to actually go dig in to answer that question, but you're right, that's a gene of tremendous interest to us in the genomic studies of Hispanic populations.
And just for the proteins in the obesity-related traits, For many of them that we do see these causal relationships, they're not the FTOs, but they're all very well-known GWAS hits. So we're not going to get all of them. It's a very subset of that 6,000 loci.
Yeah, that really speaks, I think, to winner's curse, right? So if you've got a list that you expect to see, by random chance, you'll see some and you won't see others just because our sample sizes aren't sufficiently large to robustly always detect everything that we think we'll find. But we're gratified to see an enrichment of what we expect.
Allá, allá. Acá. Acá, Fer, acá, por la doctora. Pues.
Yo tengo curiosidad de saber por qué no es importante, bueno, no sería también importante ver este mismo estudio en grupos prediabéticos o por qué el descarte también para ver la prevención.
Yes, and please don't think that we plan to ignore the type 1 diabetes as a class, or the prediabetes as a group. We will absolutely disentangle that as an intermediate step. Once we find the genes that are differentially abundant in the controls versus the cases, then we have the opportunity to see where do those genes abundance fall in the progressive pathway. Yeah, we're very interested in those as well.
And especially since insulin resistance is so highly prevalent in our Mexican American population. So we can study measures or predictions of insulin resistance in our population, which we are doing. We didn't present those data today, but we are absolutely doing that as well.
As well as looking at the quantitative measures of HbA1c as a sort of longer term measure of metabolic health and fasting blood glucose as a more acute measure of metabolic health.
Thank you very much. My name is Emma Saavedra, and your data, I really, it is like a jungle to explore. Thank you very much. I want to ask you if I guess the metabolomics is going to be more difficult to analyze because metabolism depends on the stage, on the time frame that you collect the sample. And then when you get all this metabolome data, how to put in the metabolic pathways that you are going to get from the proteomics analysis, how you are going to deal with this variability, more variability in the metabolomics data. Thank you.
I'll leave the hard part of that question to Carrie, but for the easy part of the question, we were really lucky that we had clinical research staff who were very good about collecting only fasting samples. So all of the metabolomics, all of the transcriptomics, and all of the proteomics were collected after 8 hours of fasting.
But it is a very important question. And with the metabolomics, we only have small samples at this point. We're looking to grow that, as Piper said. And you are going to get some things that are very perturbed by what you're eating and things like that. And also the sample that we're looking at is whole blood, but we're trying to expand and look at metabolomics in other tissues as well. And using that, what we showed you today, sort of like we looked in transcriptomics, we looked in proteomics, we looked in metabolomics, but some of our very smart researchers that are collaborating with us now are trying to actually put all of those things together into pathways and to understand what those data mean. And that's sort of like the next broad step that we go. And a lot of that will be done with machine learning and artificial intelligence approaches.
¿Alguna otra pregunta, camaradas? ¿No? Inge, por favor.
Thank you very much. It was a really splendid talk. I have a question regarding this highly pro-inflammatory state that diabetes and obesity cause. How is that influenced by the microbiome, which is another organ which is highly inflammatory in occasions, and especially since you can have populations, very specific ones would have very specific diets. And how does all that come together when you mix all this pool, this pro-inflammatory pool? Does that influence at all the gene expression?
So from the obesity perspective, absolutely. And the inflammatory pathways, we find a lot of what's really interesting is we try and look across tissues where we see the evidence of the transcriptomic effects and the proteomic effects. And it's really interesting because like in the bulk sequencing that we do, the bulk transcriptomics or the bulk proteomics, we don't know exactly what cell that's coming from there and what it's doing. So for example, in obesity tissue, you're going to have a lot of inflammatory cells as well. And you don't know whether it's coming from the adipocytes or where it's coming from that. So that is why what we are really working to do is to do single cell omics to try and disentangle the exact role, the exact thing that you're saying. What is it? Is it in the inflammatory cells and what is happening? And to do that, we're going to need additional tools. Those tools are very expensive, and we're pining for them and trying to build our resources for that. But it will take that kind of complex, single cell sequencing, I think, to get at those answers, or single cell omics. The other thing that's really interesting that you brought up is the diet. And one of the things that's really cool about metabolomics is that we can get information on diet because they do leave metabolic signatures. So even if we don't have great diet report data, we're able to try and look at the metabolomic data and have a better understanding of the diet. And that's actually something that Misa is very interested in doing, as she has a background in nutrition.
Thank you.
Yeah, and I'll just add from the microbiome perspective, that's really, really a fascinating area of research. Our participants at the US-Mexico border are unbelievably generous with their time and attention. They fill out survey after survey. They'll sit in the clinical research unit for 6 hours getting liver elastography, getting their heart, getting ECGs, EKGs, getting retinal scans. I mean, they are so tolerant. The thing that they don't like doing is giving us fecal samples. We just have had we have not had great luck getting people to be keen to participate in studies of the fecal matter for the microbiome studies. That said, we've tried, and I think that we have a data set now of about 180 or so participants that have given a fecal sample, and we've generated 16S microbiome data for that. And we have not yet partnered with somebody with the right expertise to integrate that data into our other omic studies, but that is certainly on the horizon.
You just answer my question.
I thought you were his.
Yeah, thank you very much, Piper and Carrie. The patient with diabetes, with many years with diabetes, they lost weight and the lipids are altered. So, do you take in account in the comparisons this situation?
In the diabetes analysis? Or
the years with diabetes that people have with diabetes, because the balance of the lipids changed radically because the insulin have an important influence in the synthesis of lipids and the storage.
Yeah, I think that's a fantastic point, because type 2 diabetes is not a static disease, right? You develop type 2 diabetes, your body is working to produce enough insulin to process the glucose from your blood. You're producing very, very high levels of insulin because you've become insulin resistant. And then your pancreas kind of burns out from that overproduction of insulin. And then you see the insulin levels plummet and your physiology really changes. But all of that very dynamic progression occurs in the state of type 2 diabetes. So studying type 2 diabetes as if it is a uniform, homogeneous state, for your system is probably wrong. And you're very much right that we should be thinking about ways to sort of dissect progression of disease and then study the omics that are relevant to those progressive states. Right now, we are just at the beginning of uncovering all of this.
Yeah, that's why to study, I think, and related to the question of the student, to study the pre-diabetics people is maybe they reflect more precisely the situation of the patient when just begin to be wrong.
Yeah, I'll also just note that through literature that I know you're very well aware of, people have hypothesized that there are different diabetes subtypes to further complicate the situation. And so getting our sample sizes sufficiently large to be able to understand omics profiles, not only at different stages of disease, robustly, but also for different types of disease is also going to be an incredibly important research question.
Okay. I have one last question probably. I think you guys are doing what every researcher in omics is doing, multi-omic studies, right? But when you showed us your Venn diagrams, I felt I've done multi-omics analysis too, and it's so sad when you come to your Venn diagram hoping that everything overlaps and you have a 0 in the middle, right? So my question here, and I think as a conclusion, it would be, what are your next steps, or what adjustments needs to be done in order for us to kind of integrate the omic analysis and find this line that we want to see, a genetic alteration, an alteration in the transcription, an alteration in the proteome. So what are your adjustments that you're going to do?
Yeah, so we were very gratified to see that those centers of the Venn diagram, when we were comparing the directly measured proteome to the directly measured transcriptome, were extremely large. They defied our expectations by an order of magnitude. Where we saw that 0 was where we were trying to use inferences that were derived from the wrong population. When we were using the GTEx models to infer the RNA abundance and then leverage that genetic data to be able to anchor it around genes. I think that what we're learning is that you can't just use white people as a proxy for all other populations. And so we've not only worked to develop bulk EQTLs that could be used to derive those same kinds of models in Hispanic populations from Hispanic people, and we also have shown that these EQTLs from Hispanic populations allow us to do better interpretations of Hispanic specific GWAS signals. So all of the evidence is showing us we need to do the GTEx work again, and we have to do it in the right populations in order to be able to interpret the genome-wide association studies from those populations. It's why Carrie and I are so excited to collaborate with Dr. Baruman and Dr. Alegre on the GTEx-MEX project, where we're going to be working to collect many of the same tissues that were ascertained in the GTEx consortium and measure the RNA abundance transcriptome-wide in this range of tissues so that we could build analogous models, but even better than that, to use, because Hispanic populations are admixed, their genomes are tiled with sources of different continental ancestry components. So you might have what you could call an Amerindian haplotype, and then followed by a European haplotype, and you can sort of paint the chromosome as this mosaic of different ancestral components. We can actually now deconvolute those local ancestry effects to be able to estimate, well, this EQTL in a Hispanic person is highly predictive of gene expression on a European haplotype in a Hispanic person, as opposed to on an Amerindian haplotype in a Hispanic person. So by reconstructing how we think about the dynamic admixture within Hispanic genomes, we can actually do better than GTEx did in the European populations. We can sort of leapfrog them and create even more accurate estimates and models that are going to work even better in this population. So we're really excited about what we're already able to do in blood from the CCHC. We are also now officially starting to collect fat samples, subcutaneous adipose, from the abdomen and the femoral area from these same participants, but we cannot biopsy their livers. We cannot biopsy their retinas. We cannot biopsy their cardiovascular tissues. And so for this, we need to have a source of tissues that are coming from postmortem samples. And so we are working earnestly with you all here at UNAM to be able to begin to do that and ship those samples up to Vanderbilt for analogous assessments so that we can provide better data that will be important for interpreting those TWAS results and making that center point not 0.
Best of the luck then.
My question is in Spanish. Can I?
Yes.
Ok, mi pregunta es, tenemos en ciertos puntos de esta investigación, me interesa bastante, no sé mucho, pero mi pregunta es, ¿puede en cierto punto de la investigación en el cual se está investigando a la gente, podemos ver diferentes formas de sus genomas, pero se pueden presentar alguno que otro polimorfismo por alguno que otro medicamento? ¿Se podría tomar en cuenta esto? Esa es una de mis preguntas. Y la otra pregunta es, ¿qué tanto podría afectar en esta parte de la investigación que tengamos algún otro que otro fármaco en el cual los resultados de la investigación pueden ser diferentes, pueden afectar en alguna parte de la transcripción, la traducción o en las proteínas que se van creando? Y el otro punto de mi pregunta es, si sale alguno que otro polimorfismo en una persona y se empieza a aparecer en cierto grupo de personas en esta investigación, ¿se puede tomar en cuenta para una base, se puede tomar en cuenta en base para comparación en otras poblaciones, en otras personas?
So there was a lot to unpack in your questions, and I love the curiosity. I think some of the things that I heard you asking was, can we take genetic information from individuals in the population and predict or look at their response to therapeutics and say, because you respond in this way, we should give you this drug. So that approach is one of the main motivating factors of this work. So that approach is called precision medicine. And basically that's why we're trying to understand all these genetic underpinnings so that we can prescribe the right drug to the right person, not based on biases of what we think a person is, like what their race or ethnicity is, but rather what's in their genetics, what their genes are telling us. Other questions that you asked is, can we look at different polymorphisms across populations and try and understand why we see, I think what you're getting is why we see different rates of disease across these different populations based on the polymorphisms that individuals carry. And if you were asking that, then yes, that's one of the pieces of the puzzle that we're trying to disentangle. Did you have other things you thought wanted to highlight?
Yeah, I also just wanted to mention that you're also right that taking drugs or therapeutics can alter the transcriptome and the proteome. And so in order to condition for that, we actually removed people who were on medications from many of our analyses and sub-analyses. One of the challenges with the population at the US-Mexico border is because they're so high a proportion of them are uninsured or underinsured, even those with type 2 diabetes are often not taking any medication at all for their diabetes. And in fact, half of the people who come in and have a blood sample for the transcriptomics or the proteomics don't know they have diabetes until we diagnose them at that visit, which means it is an unmedicated time point. Yeah.
And just one other thing to add, the metabolomics. So that's really one great point about the metabolomics is that we can actually get it. It will give us measures of what drugs people are on. So we don't need to rely on them telling us, but we can do conditional analyses. We can do additional analyses where we say, we're going to control for the fact that they're on these drugs and see what other effects we see, which is really why the integrative omics approach is so interesting.
And as a final point, I'll note that we are actually running a clinical trial right now in this community at the US-Mexico border for Ozempic, for GLP-1 agonists. So the study design is that we are giving everybody, they all start the study with prediabetes, and we are giving them Ozempic for 6 months, and then we're looking to see how their weight has changed and whether it has prevented them from developing full-scale diabetes. Then we're going to have half of them come off of the drug, and we're going to look at their metabolic memory to see whether or not they regain the lost weight or if they retain the weight loss. And we're also keeping half of them on the drug for a further 6 months to see how their weight responds for another 6 months. This is going to give us not only a lot of data about how these important weight loss drugs that are transforming obesity care across the globe perform in Hispanic populations, but it's also going to allow us to identify genetic and transcriptomic predictors of response to the drug. So not everybody responds to Ozempic the same. Some people lose a lot of weight, some people lose a little bit of weight, some people gain weight. If we can find molecular signatures for those people that are non-responders to the drug, maybe we can help pharmaceutical companies change formulations or identify secondary medications that might improve outcomes for that group. And so that's another place in which we're actively researching drug and omics in the CCHC.
Well, thank you very much. Les agradezco mucho, damas y caballeros, su presencia y me permito presentar al doctor Roberto Tapia, quien va a darnos las conclusiones.
Bueno, ante todo, muchas gracias a Jaime Berumen por invitarme. It's a pleasure to and really to envy your work and to see the dynamics that you are involved. It's just fantastic. I'm going to switch into Spanish. Yo creo que es una de las, y en verdad lo que decía la doctora Sepúlveda, de que esta asistencia, tanto presencial y espero que también en línea, de esta nueva cohorte de profesionales en la salud, tanto en el campo clínico como en la investigación, en la docencia, ya estén involucrándose en este nuevo campo y esta nueva ciencia y esta nueva abordaje del conocimiento. Cuando el doctor José Ignacio Santos y yo nos formamos, pues obviamente esto era como que Star Trek, ¿no? Era una cosa así como no imaginable. Y poco a poco nos hemos ido acercando y es mucho por lo que también comentó la doctora Sepúlveda, la oportunidad de esta colaboración entre instituciones, colaboraciones académicas, intercambios académicos. Lo que aquí Piper decía, tengo abierta contratación de postdocs o en posgrados, el programa de PSEM, al que yo formo parte también, facilita eso justamente. Así es que creo que ese es un tema importantísimo. El otro obviamente ya es la colaboración entre investigadores, porque sin lugar a dudas nosotros podemos aprender, estudiar, simular, tratar de adaptar y de incluso tener la aspiración de tener una infraestructura y un financiamiento que lo debemos de tomar como esa colaboración en proyectos conjuntos, de abordar a las grandes instancias que están en pro de estos financiamientos como NIH y hacer grandes colaboraciones, eso también ayuda mucho. Yo creo que hemos escuchado hoy aquí una película fascinante de lo que es conocer posiblemente hoy lo que es la estructura de un gen y todo lo que eso implica. Hace 20 y pico de años, cuando conocimos la publicación del primer genoma humano, se dijo, bueno, pues ya está aquí, y creo que fue el principio de abrir una puerta que apenas se le estaba viendo la primera luz que iba a llegar. Sin duda, esto nos ha llevado todo un tema de que sí, ya sabíamos que los genes determinaban la producción de las proteínas y las proteínas, los mecanismos, pero no, y me gustó mucho una gráfica que Carrie presentó que decía, conociendo el gen, medio sabíamos qué iba a pasar, pero ya conociendo toda esa cadena, ya sabemos qué pasa. Y eso es parte de lo que estamos aprendiendo. Y aquí acaban de mencionar al final un tema que para mí siempre ha sido como el punto de impacto de todo esto, que es la personalización o la medicina de precisión. A mí me gusta más llamarle personalización porque va a una escala superior. La medicina de precisión es desde la perspectiva de la alteración metabólica o de mejora o de control de una alteración metabólica. La personalización puede incluir hasta lo social y lo ambiental. Y podemos tener medicamentos muy exitosos, pero que posiblemente por las condiciones económicas, sociales de la persona, su nivel de estrés, su nivel de interacción, su falta de red social, puede que no le genere la misma respuesta de lo que podría ser para otra persona. Entonces yo insisto mucho en que veamos esa visión más amplia. Ya también tenemos décadas con los determinantes sociales y ahora hablamos de los determinantes comerciales y los determinantes ambientales. Pero ese conjunto interactúa y lo están demostrando, y de ahí el fundamento de hablar de la personalización de las intervenciones, incluso en salud pública. En salud pública, que siempre nos hemos formado con esta visión de que ante ciertas características te coloco en un cajón que por sus condiciones de variabilidad, pues las reúnes, pues ya te clasifico en un cajón. Pues resulta que tal vez de ser el cajón correcto, adentro de ese cajón hay diferencias. Y eso es lo que hoy en la salud pública debemos de distinguir. Y por eso esta motivación de hacer estudios epidemiológicos, pero con una visión de una salud pública mucho más amplia, más integrativa. Incluso las ómicas nos ayudan a esa visión integrativa de estos procesos. Entonces, creo que esto ha sido muy revelador, es extraordinario que podamos hoy escuchar digamos, este acervo de conocimiento que pues, es la punta del iceberg, porque el mundo cada vez, y tal vez estamos viviendo todos como generación que todavía estamos en estos campos, una exposición, un conocimiento exponencial de lo que sabemos y que tal vez que descubrimos a la vez que sabemos menos con todo esto. Así es que a mí no solo me queda el decir muchísimas gracias por venir, gracias por favorecer esta colaboración y de tener el espacio en sus agendas para compartir con nosotros en un ámbito académico. Y en verdad felicitar al doctor Berumen, a su equipo, esta visión que le está dando al pensar en cómo la genómica y todo la consecuencia que eso tiene puede llegar y puede tener una intervención dentro de la visión de una salud pública mucho más moderna y una medicina de vanguardia en nuestro país. Muchas gracias. Thank you very much.
Pues para terminar, agradecer mucho a las doctoras Piper y Carrie North. No nada más vinieron a exponernos sus resultados y su conocimiento, ellas nos ayudaron a organizar esto. Ellas pagaron su vuelo y la verdad se los agradezco mucho. Agradecerle a Roberto Tapia que nos ayudó también en pagar los hoteles y pues ya anduvimos rascando el resto, pero esa es la manera de organizar esto rápido y no esperarse, pedirle todo a nuestra directora, pues tiene mucho que gastar por todos lados, pero de otra manera no lo podríamos haber logrado. Se los agradezco mucho, es muy importante para nosotros. Y para terminar, nada más pasar a esta última diapositiva. Nuestro próximo simposio, ya perfectamente agendado con invitados, es el tercer congreso. Así está, pues. Es el tercer simposio internacional del estudio prospectivo de la Ciudad de México, que ni más ni menos el doctor Roberto Tapia es el líder del proyecto en México y en el mundo, porque este proyecto inicialmente lo vislumbró él con el doctor Richard Peto hace más de 25 años, y luego Jesús Alegre se incorporó y es el coordinador, digamos, del proyecto, pero un proyecto tuvo una visión de colectar esa gran cantidad de individuos, de estudiarlos, que los científicos básicos nunca pensamos más allá de 1,000, 2,000 muestras, honestamente. Ya estudiar un poco más ya era casi imposible. Solamente un epidemiólogo de este tamaño como Roberto Tapia puede ver en futuro. Y ahora hay congresos, como fuimos el año pasado a Oxford, congresos de estudios de más de 100,000 muestras. O sea que si no estás en un estudio de eso, no entras al congreso. Entonces, ahora vamos a traer a 8 personas de la Universidad de Oxford. Igual nos ayudan mucho en la organización y en el pago de esto. O sea, todo el grupo fuerte de la Universidad de Oxford. y el grupo de la UME, que vamos a presentar esencialmente pues los hallazgos, y ya el próximo año vamos a organizar ya un congreso más en forma de 3 días con más cortes de todo el mundo. Vienen después otros 3 simposios que ya los anunciaremos después. Les agradezco mucho su asistencia y muchas gracias a todos por estar aquí. Le vamos a dar aquí una constancia de agradecimiento a la doctora Carrie North. Doctor Roberto Tapia.